today we are going to do our experiment to study pollen tube germination on slide. Okay. In this we are going to germinate a pollen tube from pollen grains in the slide. For this experiment we required the materials. Okay. The materials are, you can see this is boric acid powder, the requirement boric acid powder. We need 10 grams of boric acid, we need 10 grams of boric acid. Next one, we need sucrose, 10 grams of sucrose is required for this experiment, and 20 milligram of potassium nitrate is required, 20 milligram of potassium nitrate and 30 milligram of magnesium sulfate. Okay, this is magnesium sulfate. We have to weigh all these chemicals with the help of electronic weighing machine. I had already weighed these chemicals. We can see here, this is 10 gram of sucrose, 10 gram of boric acid, 20 milligram of potassium nitrate and 30 milligram of magnesium sulfate. Okay. We are going to dissolve all these in the 100 ml of water. This is 100 ml of water. With the help of measuring cylinder I had kept it. So, this is the water, you can see it is 100 ml of water. We are now, I am going to add all these in this water. 10 gram of sucrose, 10 gram of boric acid powder, 10 gram of boric acid powder, this is 20 milligram of potassium nitrate and 30 milligram of magnesium sulfate. Now, with the help of stirrer, I am going to stir it so that it can make a uniform solution. This solution will act as a nutrient medium. We know that we need nutrients for the growth and development. For the pollen tube also we need nutrient for its growth and development. See, when we will dissolve whole chemicals in this, we are going to make a solution. This will act as a nutrient medium solution. We are dissolving it with the help of stirrer. Okay. Now, we have to keep it static for some time, okay, without disturbing, right. Now, I am going to take a slide. This is the clean slide, I am cleaning it. Okay. Now, you can see this is the flower. Flower is having pollen grains. Now, I am dusting this flower on the slide. Okay. Here I got some pollen grains on the slide. See, this is the pollen grains on the slide. Now, I am going to put this nutrient medium. Now, this is the crystal clear nutrient medium. I am taking one drop of this nutrient medium on the slide. Okay. I kept one drop of nutrient medium here. Now, with the help of cover slip, okay, I am going to prepare this cover slip. Okay. I am going to keep this cover slip on the slide. This slide is having all the nutrient medium as well as the pollen grains. Yeah, this is the perfect slide. Okay. I am going to drain out the excess of the nutrient medium. देखो filter paper की help से excess of nutrient medium को हम जो है remove कर देंगे. Our slide is having, now this is the perfect slide which do not have any excess material in it. It is having slide as well as the nutrient medium as well as the pollen grains. Okay. Now, we are going to see this slide under the microscope. Now, we can see under the microscope, okay, under 10x low power microscope, we are seeing that many pollen grains are visible. Okay. Pollen grains are showing its two structures, that is entine as well as exine, right. Now, I am going to keep it under 45x. Okay. You can see under 45x, this is the clear structure of the pollen grains. After half an hour, we have to see a small opening arising from the pollen grains. That opening will be the starting of pollen tube. So, again after 30 minutes, we have to see this slide and we will be able to see the number of pollen tubes arising from these pollen grains. Okay, students. So, after 30 minutes again we are going to see this video. Again students, we are seeing our slide after half an hour. We will be able to see a very small tube opening arising from the pollen grains. Okay, now under the microscope, first we had seen that it is having an outermost covering exine and entine. Both of these parts were visible under the microscope. Okay. After half an hour, we will be able to see pollen tube arising from this pollen grain. Okay. A small pollen grain is arising the pollen tube. Not all the pollen grains which we had dusted on the slide will able to produce the pollen tube. Okay. So, we have to count the number of pollen tubes arising and total number of pollen grains present in that particular area. Okay. We have to count the number of pollen grains and note it down. Okay. So, we have to observe, the final observation will be arising the pollen tube. With this, our experiment to study pollen tube germination on slide is being completed. You can note down your observation, you can draw the diagram, and at last you can conclude your experiment. Conclusion is the arising of pollen tubes from the pollen grains under the nutrient medium. Okay students, then thank you. Please note it down in your copies.